The last speaker is Dr. Anton Nekrutenko, the global system of data analysis in a public scientific cloud.
Thank you. Thank you. Thank you very much for inviting me. So this is sort of a Russian translation of what I'm going to be talking about. I think I invented the term public note, публичная научная облако. Well, it's okay. So it's a global system for data analysis, for cloud-based system for data analysis, and SARS-CoV-2 as the engine that drives the development. Throughout this presentation, there will be these URLs. You can go to them and sort of see what I'm talking about. If it's possible, I sent the link to this presentation to Yulia, and if she can share it with them, so it can be shared if anybody's interested. Okay, so I come from Kiev some time ago, and then I moved across here. Well, actually, right now I'm closer to Moscow than US, but anyway. And right now I'm at Penn State. So at Penn State, we run a project which is called Galaxy. It's kind of there is a history to that name. And so I'll just briefly explain what this is. So this is it's many things. It's a software framework which is designed to allow people to analyze very large collection of samples via UI. You can do it through API as well, of course. But it's really the only place where you can go and analyze hundreds of thousands of samples through user interface. It's a very hard problem, and in general, and so we have, I think, a good solution for this. And because modern sequencing is cheap, very few people sequence just a few samples. Most people sequence hundreds, perhaps thousands of samples. And in the title of this talk, I was trying to say that SARS-CoV-2 is sort of an engine of progress. And it's because it's the first pandemic which coincides with relatively cheap sequencing. So it's the first time that we see data sets of this size. So it's a good test for many software tools. For example, how do you exactly build phylogenetic trees when you have 1.5 million sequences? How do you do selection analysis when you have many sequences like that? How do you reliably monitor emergence of new variants where you have continuous stream of sequencing data sets and so on? And it's actually a lot of software which we considered good before doesn't really scale. So it's an interesting problem. And so in that sense, I guess SARS is a good thing, scary thing to say. So it's also a workflow system. So essentially it's an execution system which allows you to run tools on data. So we have workflow language. This is, for example, this is an example of one of the SARS workflows that I'm going to be talking about. And so it's also engineered to run on latest modern high-performance compute infrastructure, and that includes public and private clouds. So we have some instances which run on Amazon, some projects where we use Google. However, really the subject of this talk is about free system which uses public clouds. And this is very important because when you talk about compute clouds, you usually think about Amazon or Google or Microsoft Azure or other smaller systems. But the problem with these clouds is that they're wonderful for doing, for example, many types of data analysis problems that we have in biology, but they're not free. And if you live in the US, for example, you work at the US university or at a research institution, you perhaps can pay for that. But if you're, I don't know, in Russia or Ukraine or somewhere else, even in France, for example, it's difficult for many reasons. Some of the reasons are purely protectionists. These are American things. So for example, Europeans are much less likely to use them. And some things are just paperwork stupidity of this sort of that how do you reconcile the bills. So so why we have a lot of systems which rely on commercial clouds, it's not a good solution if we're looking to give the entire world an ability to analyze large data sets. And so there are several types of free public clouds that exist. So in US, there is a system called Exceed. It's essentially a network of multiple supercomputing centers, such as TAC, which stands for Texas Advanced Computing Center. There's also Jetstream Cloud. These are all projects funded by National Science Foundation. And so I mentioned that Galaxy is software. So this software can be used to run Galaxy instances. These are actual websites where you go and analyze your data. There are many instances, but there are three big ones which operate in, this is a large collaborative effort. So one in the US, one in Europe, and one in Australia. And they all rely on public clouds. In US, that's Exceed that I mentioned. In Europe, it's part of the German network for bioinformatics infrastructure, also Elixir, which unites a number of European countries and also provides infrastructure. And in Australia, it's Australia's specific Nectar Research Cloud. So these are all examples of free public clouds that researchers can use, and they can use them across the boundary. So if there are kind of doctors without borders, these are clouds without borders in some sense. There's also a number of local instances, for example, through Europe. We don't really track that, so Galaxy doesn't call home. So there are hundreds of instances, but these are the ones that we're actively working with as well. What I mentioned so far, it's a software. You can install that software. It would allow people to run open source, well, actually any tools. We, of course, in public instances, we only use open source tools, but there are proprietary Galaxy instances which use proprietary tools. And so you cannot do any analysis without tools. And Galaxy embraces, we have tight relationship with Bioconda. It's probably the biggest effort to simplify installation of open source bioinformatics tools. It's essentially a dependency management system, but it's a version dependency management system. So if you run some old version of, I don't know, BWA, and you want to rerun it 5 years from now, you can do that because all the versions are preserved here. We're also collaborating with Biocontainers, which is an effort to containerize all these tools. And so again, to kind of standardize their execution. This is all about making data analysis reproducible, predictable as well. So this screenshot here, this is our app store. It's called Galaxy Toolshed, and this is where all global Galaxy instances pool their tools. But that system relies on Bioconda for dependency resolution. We also have comprehensive training network. It's a community project. If you remember the first slide, it had this cloud of different organizations. It really spans US, Europe, Australia, Africa. So there are a very large number of contributors. And so we developed these training materials, and they range from sort of basic, how do I analyze the data, to more of the more complicated things, such as, for example, examples of particular types of machine learning analysis and so on. And we run a number of webinars. This slide is again focused on SARS-CoV-2 because that's the game of the day. We even published a few papers about how to teach during the lockdown, because what else to do? So motivation behind our SARS effort is this. If you look at the landscape of coronavirus data analysis, it's not better than when you read the news from different places. It's all very confusing and it's hard to know what is from where. So at the research level, at our level, it's unfortunately the same. So SARS-CoV-2 data analysis is very fragmented. You have large institutions that know what they're doing, And so they're performing reasonable types of analysis. These institutions, probably the best effort is the one in the UK, called UK effort. That's the group of people that know what they're doing. But it's a power law distribution. So you have these large groups, then you have all sorts of smaller groups in different countries, and then it becomes a little bit more complicated. And it's not to blame anybody, it's just imagine yourself in the shoes of somebody in, I don't know, in Tunisia, trying to process samples from a local hospital. What do you exactly do? What kind of system are you going to use? Well, again, I just mentioned the problem with the clouds. So if this hospital generates a lot of data, you don't really have infrastructure. But even if you have infrastructure, how do you call the variance, for example? And those of you who are listening, well, who are still listening, it's 6 p.m. Calling variance is not particularly complex problem, but in the past 10 years, most of the effort was because of the efforts such as 1,000 genomes and other large human pangenome, all these efforts. We know how to call variance in humans very well in deployed systems, but the simpler actually haploid mixtures such as SARS-CoV-2 were they're kind of less familiar to the wider cloud. So as a result, we have plenty of papers that use, for example, GATK for SARS-CoV-2 variant calling, which is complete nonsense. But I mean, GATK will call variants. So you will get obviously lineage-specific variants from GATK. But if you're looking at interhost variation, if you're looking for cases, do you have co-infections, for example, then that's not a good approach. And most of the current analysis are focused on generating consensus. And so you're a hospital, you sequence the patient, you generate consensus, you compare against GZ, and you say, okay, well, this is some kind of a derivative of the Delta variant. But that sort of a fine grain, what's happening within individuals, that's ignored. And as I mentioned, the access to infrastructure varies from country to country. So our goal here is to develop a system which can be used by anyone. And so what we've done, we've developed a series of robust workflows. These are validated workflows. They're developed as using proper software development practices. They have tests. They're deposited, they're versioned, they're deposited into proper repositories. And we'll talk about this later. So most of the COVID data comes in the form of amplicon-derived sequencing using RTIC protocols or other protocols. And it comes primarily from two technologies, from Illumina and from Oxford Nanopore. There's some small number of data coming from other technologies, such as Ion Torrent or PegBio, but mostly it's Illumina and ONT. And so as a result, we developed workflows for ONT for RTIC data and for Illumina for RTIC data, and also for metatranscriptomic data. So this is the rare types of studies, there aren't that many of them, when actual samples from a patient are taken and RNA is isolated, and so it's done using RNA sequence. So there is no amplification step, and that's ideal data for looking at intra-host variation. So 4 workflows: Oxford Nanopore for RTIC, Illumina for RTIC, and 2 kinds of Illumina workflows for metatranscriptomic data. And then we also have a reporting workflow which generates outputs which can be used for interpreting these results. And we use, so this is designed to be run on hundreds and thousands of samples, and we operate it in 2 ways. So on one hand, we have public, so anybody who would go to this covid19.galaxyproject.org can run these workflows now. If you have your own data, go ahead and run them. We also run them in an automated way for all the COG UK data, which comes in also for data coming from several countries, which use this as their primary analysis framework. And then we generate dashboards. And for those of you who are not familiar with the notebook environments, probably most people familiar with Jupyter, or our studio, but there are these wonderful new things that are popping up, and one of them is observable, which is a JavaScript environment, but it's an amazing reactive way to represent results of your analysis, and I'll show an example of that. So workflows that we develop, they're deposited into, there are two kind of a global hubs for the workflows. On the US side, it's DocStore, run out of UCSC, University of California, Santa Cruz. It's part of the Anvil project and many large-scale projects on the US side. And on the European side, there's Elixir effort called Workflow Hub. So this is how we used to, this is the outlets we use to distribute our workflows. And well, in the end, this is just, this is data like this. So those of you who used to look at PCF files, we don't, we provide various kinds of outputs, but we keep this all. So we keep the primary data, we keep all the intermediate calls, we keep all the consensus sequences, and it turned out to be extremely useful because if you rely on, for example, SRA or usual places where sequencing data gets developed, sometimes things get deleted, but we actually keep everything from original read level data to all the intermediate calls to consensus sequences, and this is again for hundreds of thousands of samples. And we distribute this data into three primary ways. We are partnering with Viral Beacon, in Barcelona, which is, we also use the FTP site at Viral Beacon to distribute all the raw data, which I just talked to you about, the read level data and all the variant calls. So if we want to go back and examine this, that can be done. And we also provide these interactive notebooks. And so they look like this, for example. This is an example for Irish data. So Ireland is using us for processing of some of their data sets. So once analysis are completed on the Galaxy side, they can then ingest it into this observable environment. This is this new JavaScript environment I just mentioned. And it's really, you can build amazing widgets here. So for example, let's recompute for a particular slice of dates. You can do, and you can add these widgets as you go. So we have several types of widgets here, just the visual representation of data, variance per gene, the lineage distribution, and also there is an ability to look up individual variants, for example, how the frequency of individual variants tracks across time. So you can just select variant here and do that. So well, that's not particularly interesting one, but there's a number of others. So, well, you get the idea. Also, we keep track of these variants of concerns, the new lineages which continuously occur. And this, for example, this is all the Delta variant sites. And again, you can track, you can see where they're beginning to appear in the data and various kinds of reports. And again, it's open. You can clone, you can fork this, you can add widgets here, and so on. So as I mentioned, coronavirus was a wonderful thing in terms of scaling up software. So as I mentioned, we run primary 3, plus we have many of the regional instances in Europe primarily. So France, Belgium, Estonia, Spain, Italy, and many others. And so they're doing their individual things, but they all have access to the same infrastructure. They all have access to the same tools. This is done through so-called Pulsar network. Pulsar, it's a system for running jobs, running compute jobs on remote resources, which is developed as a part of Galaxy. It's a system which was also developed by us. So you can imagine that the entire world of computational resources, and these resources only need to run a Pulsar to appear as a Pulsar node, and then any Galaxy instance in the world, whether it's in the US, in Estonia, in Poland, in Greece, in Crete, can schedule jobs on that node. So for large efforts, for national efforts, for COG UK efforts, of course, this is all done automatically. So we have an automated way to trigger these workflows, which I showed you in graphical interface. So we have a way to trigger these things programmatically. And then we keep all the data. This is an example of JSON file with batch IDs, history links, individual accession numbers for COG UK data. This is all stored in Barcelona. So on one end, we're providing dashboards, so something for a general public to consume, as well as we provide all the other information for reanalysis, re-examining things, and so on. And you can do a lot of interesting science in this, unfortunately, which I will not have time to talk about. So that's sort of a summary, is that we take data from national resources. For example, COG UK data comes from European Nucleotide Archive. In US, data comes from SRA. Then we use our workflows, which are in the workflow hub and in the doc store. This is where national Galaxy instances get the workflows. Again, they're versioned. And the outcomes of running these workflows are distributed via, so everything can be obtained as raw data or can be consumed as ready visualizations. So you can, again, if there's a way to send the URL for this presentation to all the listeners of that talk, so these things here are the live links. So you don't have to trust me, because in half an hour, it's very hard to describe complex software system. Normally, I would do a hands-on demo, but it's hard to fit this in half an hour. So maybe what I'm telling you is complete nonsense. Maybe I made all this up. So you can go ahead and try this. For example, this is an example of hands-on tutorials which we develop. And this document describes everything. So you go to, it explains to you that you need to go to a Galaxy instance like this. You can do it in the US, in Europe, in wherever you are. It provides sample data. So this is the set of Arctic data sets. So you can play with this data. You don't have to use this data. You can substitute this with the data sets you actually want to analyze. And then it goes through all the steps explaining. So it's a step-by-step description of this analysis with explanation of all the intermediate data sets. There are a lot of things which I didn't have time to mention, which these workflows generate. For example, variant frequency plots, which are good for looking at limited number of samples. There is also this page, which summarizes our effort and explains how our resources can be used. This is the link to the current dashboard, such as the one that I showed you. So it's the same thing as this. The one thing to remember about observable notebooks is that because they're JavaScript driven, they're actually executed in your browser. So this is a kind of interesting serverless way, because if you were doing this in a standard notebook environment such as RStudio or Jupyter, you need to have a server and a client system, but here everything happens in the browser. And this is the link to the viral beacon, which is, let it load. Which stores all the data from all the samples that we process. And there will be publication coming out in October describing this. Okay, so at this point, let's see, questions.
Okay. One question. Do you think that the evolution of resources will go to the aim of more and more open source resources or more and more payable resources?
By resources, you mean clouds or tools?
Yeah, both clouds and tools and their content also.
Well, the clouds, it's a war, obviously. I mean, there are. I mean, it's hard to predict this, because obviously public clouds that I mentioned, they rely on scientific funding. So they're usually funded by scientific funding agencies in respective countries. But again, that's another kind of a weird effect of COVID. It was good for that funding. So for now, these resources aren't going anywhere. In terms of tools, open source is really the only way, because otherwise, how do you know what it does? So, I mean, if you have a variant caller and it does something, well, maybe you don't care. I don't know, but I don't think it's possible to evaluate results based on black box magic. So, but it depends on a particular field. In genome analysis, in particular, almost everything is open source. It's a kind of accepted practice in the field. In other fields, it's not so straightforward. For example, in proteomics and mass spectrometry, in image processing, that's not as clear. And if we're talking about the future, probably image analysis will be kind of the next big data wave, because sequencing is it's kind of a stabilizer. There will be a lot of sequences. But with these new techniques, as different kinds of microscopy, the cryo-EM, all these things, there will be just enormous explosion in the need for image processing. But there is also a lot of open source development happening here. Of course, I'm very opinionated here. We don't run anything which isn't open source, and we don't need to, because once new technology appears, there will be a number of new tools. Of course, you also need to know, which tools are good, which are bad, but that kind of it can be easily judged from GitHub repositories for these tools. So open source really allows you to gauge things. It's like, в Булгакове осетрина второй свежести. You can tell первой свежести осетрина или второй based on GitHub traffic.
Okay.
So, Nikolai, can I ask? So, Anton, thank you for accepting the invitation. So, I first heard about Galaxy like 10 years ago, maybe even more, being still a postgraduate student, and it was still already impressive. And now, 10 years after that, it's more impressive. But what is your foreseeing, or like next 5 years? What will you be investing in the frame of Galaxy, or maybe it will be new products? So, where this development is going? So what is kind of far and near goals for that?
К победе коммунизма. It's hard. Unfortunately, it's hard to think in 5-year terms. But it's hard to sell free stuff. So it's a sort of a constant battle in terms of, for example, these workflows that we are describing here. I mean, they're used a lot, but it's sort of like with vaccination, right? there's some reluctance on using good open source stuff. So there's some people think that if you buy something from CLC Bio, that will be better. I mean, it's okay. I mean, but it's kind of the same thing as with vaccination reluctance. It's very similar. So I guess my plan for the next the plan for the next 5 years is to, well, there's a roadmap. Of course, we need to make interface more robust for really large scale. We need to, and we're working on different ways of representing complex samples, and make completely detach sort of the instance from compute, so it can compute anywhere. Use GPUs. There are a lot of things that we need to do.
Thank you. Anton, thank you very much.
Thank you. Thank you for inviting me.